coli is based on T7 RNA polymerase. And so T7 RNA polymerase is a really robust RNA polymerase that makes a lot of transcript when you turn it on. So the way that this works in the cell is that there's E. coli cell, and in that strain on the cell's chromosome itself is encoded the T7 RNA polymerase. And on the chromosome there's an operator and a promoter, and it's engineered so that there's a repressor ahead of the T7 RNA polymerase called the lac repressor. So on the chromosome, without lactose or an artificial lactose called IPTG, there's a repressor that turns off the T7 RNA polymerase. So if you just grow up the cells and without lactose, T7 RNA polymerase is going on. So that's one element of it. So we'll talk about what happens when we turn on that gene first. And then you have a plasmid, and if you transform E. coli with a plasmid that has your gene of interest, and now a T7 promoter. So the plasmid itself isn't going to do anything until you turn on the T7 RNA polymerase on the chromosome. So what you do is you grow up the E. coli, and we'll talk about the growth phase in a second. But then you add lactose or IPTG, and this is a molecule that will actually bind to the lac repressor and change its conformation, and the lac repressor will leave the operator site here and turn on the T7 RNA polymerase gene. So then, when the T7 RNA polymerase gene is turned on, it'll find its promoter on the plasmid itself, and then express the gene that's on the plasmid. And so it's kind of a two-step thing. The T7 RNA polymerase is turned on the chromosome, and then it finds its promoter on the plasmid and makes RNA that gets made into protein. Yep.
Can you have it so that the protein you care about is secreted, so that you don't have to then lyse the cells?
Yes. So there's sequences that you can add to this to export it from E. coli cells.
So then you can keep using the same coli, right?
In theory, yes. Sometimes it works and sometimes it doesn't. Sometimes that little secretion signal gets stuck in the membrane or between the membrane. In E. coli specifically, there's a popular way of actually exporting it between the two membranes. And in yeast, there's a common secretion pathway that's monitored. Another question? So this is kind of the workhorse here. People spend a lot of time trying to figure out how to trick E. coli into making. Our gold standard was let's trick E. coli into making 20% of its total protein as our protein of interest. Okay, so that's a pretty high bar. Yep.
Why the E. coli does not die due to the toxic effects of such an.
Well, yeah. So that's sometimes. So we want to try to express as much protein as possible without it dying, and that's the trick. so in every part of the challenge, and Biolab sells hundreds and hundreds of proteins. Part of the challenge is each protein is special, each protein is different. So so that brings me to this particular graph. So this is a bacterial growth curve, and so bacteria, when they're growing, there's what's called a lag phase where it's just kind of waking up, and then this log phase is when all the systems are working most efficiently. Transcription, replication is happening at maximum, and then usually something runs out, usually a nutrient runs out, and it goes into the stationary phase, and then the cells die. So when we think about expressing a protein, we want to turn on that T7 RNA polymerase to turn on the expression of our genes right when the cells are really happy and the metabolic, all the metabolic pathways are happening at maximum efficiency. So typically we turn on the gene in log phase. It produces a lot of the protein, and then we really don't care afterwards if the cells are starting to die. So it's right on the edge. We want to try to maximize the protein expression without killing the cells. So it's a bit tricky, and there's a lot of people who spend a lot of time trying to figure out exactly those conditions. So, but to your point, is how do you express a toxic gene? So you can imagine like a restriction enzyme that cleaves DNA or a DNase enzyme that targets DNA for destruction. It's really hard to actually make in E. coli, right? So if you have this protein that once you express it, it's going to kill the cell. That's a real challenge, right? So there are a couple different strategies for producing a toxic protein. One is to make sure that this expression is off completely until you turn it on. So there are different strategies and different repressors that you can actually have tighter binding and turn that gene off until you're ready to turn it on. So if you grow up the cells and the gene's off, the cells grow in exponential phase, and then you turn on the gene, it'll make the protein, and then maybe kill the cell. So it's again that balance of expressing enough protein before the cells die. So really you want to express it, maybe in the late log phase, so you have a lot of cells. It gets turned on, kills the cell, but you have a lot of cells that are making that protein. So this is a common challenge, and again, there's a lot of time spent on trying to figure out the best growth conditions. And as you can imagine, with proteins from hot springs, those proteins work at 75 degrees, right? So if you're working with a protein that's a little bit toxic, you can actually grow the E. coli at lower temperatures and just reduce the activity. So if you like kind of optimizing things and figuring out these puzzles, like this type of expression profiling is super important. Doesn't get the praise that it deserves, but it really sets everything else up. Okay, so once you've figured out the conditions, the temperature, when you're going to turn the gene on, then you transfer that over to these high density fermenters. And in these fermenters, again, another team of people really figure out how to keep the E. coli happy, how to keep the pH from changing, how to keep the oxygen levels right at the optimal level, to really make these E. coli happy, so it's expressing a lot of your protein. We started off, with these small scale, and then scale all the way up to 1,000 liters. So really, 1,000 liters is really the maximum that that a company like BioLabs could do for fermentation for the E. coli. Other companies, a lot of therapeutic antibodies have massive fermenters that they secrete antibodies and that sort of thing. So they have massive fermenters, but it's essentially the same sort of process, really optimizing the growth of the cells, optimizing the expression and inside the cell or outside the cell. So there's a lot of meticulous work that needs to happen for that to be efficient. And then the fun part of purification. So I always like this part. And just back to my roots in the thermophilic proteins. The first step that we'd always do is our proteins in these E. coli, the proteins that we were purifying, would be thermostable and really able to withstand boiling temperatures. So we take all our E. coli, we put it in a boiling water bath. All the E. coli proteins would be natured, and we centrifuge all the junk out, and we do that with our thermostable protein. So we were really spoiled. But in the real world, what happens is that the cells get lysed, broken open, and so you have thousands and thousands of proteins. And really the trick is to separate the proteins that you're interested in from all the other proteins in membranes. So these are examples of columns, and you pass the proteins over the columns, and the columns differ by charge, by chemistries. And so as the proteins pass over the columns, under certain conditions and certain pHs, some of the proteins will stick, and some of the proteins will flow through. And then you're left with an enriched pool, and then you might do it again with a again, this is a whole area, and people specialize in trying to figure out purification, so all of these different columns and purifications. And again, the challenge is that you have hundreds of proteins that you're working with and selling. Each protein is special and each protein is different. There are a few tricks there. Some people use what's called a his tag, and so that's just adding 8 histidines to the end of your protein. And histidines have a very specific charge that will bind specifically to a nickel column field. So it's a really nice way of, in a single step, capturing proteins on a single column while everything else flows through. So I find this step super satisfying. And what's interesting is you can do this on the small scale, and then these bigger columns have like 10 liters. So that's the size of purification. Okay. So any questions about kind of the manufacturing process? And I should mention then, once you have purified proteins, then there's other teams of people who check the activity, check the quality control. So that's what you're doing, Josh, right? Design the assays to make sure that this is the protein that you think it is, and there's no other contaminants that might hinder the reaction. So again, that's a whole other area that's super important for. Yep.
So for part of that, for enzymes specifically, they do it the whole
Yeah, right. So we have the enzyme that we purify, and really development, you want to see what the maximum activity is in different buffers, and find the right buffer for that enzyme. So that's certainly a critical step of that comes kind of upstream of that to find the useful activity in that. The bigger problem is that there are these contaminating exos from contaminating enzymes from E. coli that really cause havoc with DNA enzymes. So like exonucleases tend to chew things up, and they're really good at that. So even at a very, very slow level that you can't see a protein go, they see the activity. So you might have to say, okay, we have some exonuclease contamination here, we put to another column. It doesn't have to see. So and the interesting thing is that customers keep pushing the limits of how they're using these enzymes. And so enzymes that were pure enough 5 years ago are not pure enough now. And right to the fact that people are trying to use enzymes to look at single molecules, and that's a really, so if there's any sort of contamination, they're going to see it as single molecule reaction. So it's a big challenge. I think it's a constantly evolving thing. They're constantly trying to push it. So we talked to Josh about that. Yeah, that's a good question. Okay, so we're going to move on to looking at enzymes in action. So now you've kind of thought about how to discover a new enzyme for an application. You know how to clone it. You know how to manufacture it. And now you want to use it in an actual reaction. So this is my favorite enzyme. This is a movie of a DNA polymerase. And so, what you see here is the crystal structure. This is the DNA that's being extended. This is the nucleotide that's coming into the active site. And these are what's called the fingers. So in the structure of a polymerase, they call it like a hand structure. So the DNA itself lays in the palm, and the thumb domain helps grip the DNA. And then these fingers domain, a nucleotide comes in to the active site, and then the fingers close. And if the nucleotide is properly base paired, the nucleotide will get incorporated, and then the DNA will shift by one base pair. But what's kind of cool about these polymerases is that if the wrong nucleotide floats into the active site, the fingers will close, and the polymerase senses that, wait a minute, this is not the right geometry. And so the fingers actually open again, the nucleotide flows out, and then hopefully the correct one will come in, the fingers will close, the DNA will shift by one nucleotide. So as you think about your PCR reactions that you've done in the course, like this is what's happening in the test tube at about 1,000 nucleotides per minute. So this is an incredibly efficient, really, machine that's sensing the right nucleotide, incorporating it, and then moving on to the next one. It's kind of amazing. So there are two flavors of DNA polymerases. The first one is Taq DNA polymerase. This is a polymerase that has a 5' to 3' exonuclease, and so if it's synthesizing along and it encounters a piece of DNA in front of it, it'll actually chew off that piece of DNA, and that's a really important feature for qPCR. Taq polymerase is really, really robust. It's used in a lot of diagnostic tests because it just works. It's really not sensitive to a lot of inhibitors, and it just works. So it's really the backbone of PCR for a lot of applications. The downside of Taq DNA polymerase is that it's low fidelity, meaning that it doesn't have a way of proofreading. If it puts in the wrong nucleotide, it'll just keep on going. So if you have a diagnostic test where you're just looking for the presence or absence of a band, it doesn't really matter, right? So a few mutations here and there won't really matter. But if you're cloning a gene and you want to have completely good fidelity and amplifying whatever you amplify, and to have confidence that whatever you amplify is going to be the same sequences that it started with, you need a high fidelity DNA polymerase. So this polymerase actually has a proofreading exonuclease that we'll talk about in a second, and it's very high fidelity. So examples of that are P5 polymerase, PFU, fusion. They go through other names as well. Yeah. So I think this is an amazing feature of these polymerases. So in these proofreading polymerases, there are actually two domains in the structure. So this purple domain is the polymerase active site. So that's responsible for incorporating nucleotides. And then this blue domain is what's called the proofreading exonuclease domain. So it's a physically different part of the protein. And so when the fingers close in these polymerases, and if it incorporates the wrong nucleotide, the polymerase itself will actually sense that it's the wrong geometry, and it will actually physically send that wrong nucleotide to another part of the polymerase, this exonuclease site, where it gets clipped off, and then the primer strand goes back into the polymerase site and keeps on going. So it's kind of amazing to me that the polymerase itself can actually sense that something's wrong, send it to another part of the protein, deal with it, flip it off, send it back to the active site, and keep on going. Right? So this is, in our own cells, this is happening also, right? So when our own cells divide, we want to make sure that our chromosomes in our bodies are copied correctly. So the polymerases in our bodies have proofreading exonuclease, and some cancer mutations actually are in this exonuclease site that causes more mutations to occur and tumors to proliferate. So it's from the bacteria to the human, but there are lessons in the replication as well. So I think that's a really cool feature of polymerase. So why does it actually matter? Well, having a sense of polymerase error rates kind of in context of the real world. Did anybody fly Air Portugal here? You did?
Yeah.
Did your bag come with you?
No, it was one of the 97.
Really? Okay, you did, okay. Well, so Air Portugal, they lose 3 bags per 100, so a 3% error rate. British Airways, anybody fly British Airways? Did your bag come with you?
Oh, yes.
Okay, good.
Always.
Good. So they have a 2.5% lost bag error rate. JetBlue does a little bit better, although I've lost a bag in JetBlue, so it's 3 bags per 1,000, so 0.3% error rate. You look at lost mail, according to the internet, the U.S. Postal Service loses 3 pieces of mail cell for every 100, so a 3% error rate. Typos, the average typo rate is 10 typos for 100. Does that sound about right?
Yeah.
No. Yeah, I think, it depends on the person, but that's about right. So, you know, if you look at polymerases, the numbers are pretty impressive. So even TAC polymerase that doesn't have a proofreading domain, it only makes one mistake every 6,400 nucleotides that's active. So if you think about that movie, the polymerase is opening and closing the fingers, sensing the right or the wrong nucleotide. And most of the time, even without a mechanism to proofread, it's sensing that correct geometry and incorporating the right nucleotide. So TAC polymerase makes a mistake once every 6,400. Q5, because it has that proofreading exonuclease, only makes one mistake every 1.8 million base pairs edits. That's amazing, right? So it's an incredibly fine-tuned machine for synthesizing nucleotides. Okay, so how does that really affect the real world, right? Yep.
So that's a very impressive number, but there's 3 billion base pairs, so there's roughly 1,000 mistakes every time a cell divides.
Yes.
How
Yes.
does that work?
Yes, that's right. So every time a cell divides, there's mistakes. So all organisms have really robust DNA mismatch repair pathways, which are super important, right? So the polymerases are only so good. Mistakes do happen, and like anything else in life, is how you deal with mistakes. So in the cells, they're a really robust mismatch repair. So their repair systems are scanning the genome and are associated with the replication machinery to look for mistakes. And those mistakes really stick out in DNA. If you picture that beautiful double helix, the mistake really looks different, right? So there's really robust mismatch repair systems. And again, that's another area in cancer where there are mutations in the mismatch repair systems in cancer tumor cells that help, that cause more tumor genesis and more proliferation. So that's the question. So in molecular biology, why is this important, right? So if you're trying to amplify 1,000 base pair PCR products, taking into account those numbers, with 30 cycles, which is a lot. You use TAC polymerase, about 2/3 of those PCR products will actually have mutation. If you're trying to amplify 3,000 base pair PCR products, 100% of them will have at least 2 mutations. So again, TAC is robust, get a product, but if you're trying to clone a gene and have that gene be what you started with, you probably wouldn't use TAC, right? So Q5, on the other hand, if you have a 1 kb product, 99% of the things will be right, and a 3 kb, 96% of the PCR products will be correct. So if you're cloning a gene, use a proofreading polymerase, right? So you have a better shot at getting the right ones. The other thing to keep in mind in the practical sense, the more cycles you have in the PCR, the more base pairs you're synthesizing, and there's more of a chance for errors to be incorporated. So if you wanted to use TAC for a 1 kb PCR product, if you just simply lower the number of cycles to 20, then you have a better shot at getting the right PCR product there. Okay, so there's other types of PCR that are really important. For diagnostics especially, there's a real movement to do PCRs directly from tissue. So right now, if people are trying to do molecular diagnostics from blood, from urine, from poop, they don't want to purify the DNA from those tissue samples, but they want the PCR to work directly with the samples. So in tissue samples, there's a lot of inhibitors that really screw up PCR. And so what some researchers did is they actually take the wild type TAC from that hot springs in Yellowstone and then make mutations so it can work directly in different types of tissue samples themselves. Okay? So thinking about patents, right? This is a non-naturally occurring mutant polymerase that can do this particular task. So this would be that. So it really simplifies the workflow. It's cheaper, faster, more robust. Multiplex PCR is another method that instead of using just a single foreign reverse primer, people actually optimize the reactions. In this case, there's 15 amplicons all at once. So if you're looking for the presence or absence of a band, instead of doing 15 different reactions and 15 different gel lanes, you combine all the different primer pairs together. So this is really popular for like looking at trying to amplify 15 different cancer targets or cancer genes and then sequencing them. And that this has been pushed to hundreds of targets all at the same time. Yep.
Can you talk a little bit more about direct PCR?
Yeah. Yes. So it's typically like from a blood sample, there's a lot of inhibitors in blood and also in the tubes where you take blood. There's UTA, there's citrate, there's other preservatives that will preserve the blood for the chemistry, but we want to analyze the DNA. So the polymerases themselves don't like UTA or citrate or some of the preservatives in blood. So if you try to take a drop of blood and put it in a tube, you do PCR to see if there's a gene there or an infectious material there, it wouldn't work. So typically people take that blood sample and then they do a DNA purification to get pure DNA and then do a PCR. So this instead of doing that DNA purification step, you skip over that and put the blood directly into a PCR reaction and then do the PCR itself.
I
No.
also heard that there was some sort of PCR that you could do on slides. Is that the same?
Probably, yeah, probably. So it's have tissue and then yeah, probably. So yeah, so it's a challenge for sure, and there are inhibitors everywhere. Even in plant tissues, there are inhibitors that inhibit PCR. Okay, so this is another flavor of PCR called digital PCR, and the concept here is that it's a quantitative way of detecting a certain amplicon or target. Okay. So what it is, it's simply PCR. So you're looking for amplification of a particular target with a forward and reverse primer. And except that instead of using a test tube to do your PCR, you're actually creating these little tiny droplets. And in each one of these droplets is the forward and reverse primer, the polymerase, the nucleotides, the buffer, and also typically a dye. Okay? So if this droplet contains your target, PCR will go, right? So the forward and reverse primers will yield to your target. The PCR will happen and create a whole lot of DNA, and that dye will bind to the DNA that's just made. So here in this droplet, you have your target. So this droplet is the target. Contains the target, you'll get a PCR, and the dye will interpolate here, and you'll get a positive signal. If in another droplet there is no target, then you'll get no PCR. And what this digital droplet PCR reader keeps track of, it says how many of these droplets have the target or don't have the target. So if you're comparing, two different samples, you can actually count up the droplets and get a digital readout of how many targets were in that sample. So you have 14,000 in one sample, and you might have 2,000 in another. And simply doing PCR the way we know how to do it, it's just counting up droplets, so. So the cool thing about this too is that, again, would you use TAC in this situation, or would you use, limits? Why?
Lower resolution, you don't need the resolution.
Yeah, we don't really care about the sequence, we just care about is it there or is it not there, right? So, but what's interesting here also is that it's not just garden variety TAC, because PCR in a droplet behaves differently than PCR in a test tube. So they actually had to engineer the TAC to make it work really well in a droplet rather than a test tube. So there's all these incredibly creative ideas that get squashed because it doesn't work, and then you have to engineer the enzymes to make it work. Yeah.
So droplet is one PCR reaction? or
Yes.
several?
Well, so each droplet contains all the components for doing PCR, and the droplets will contain it will contain DNA, and your target will either be in the droplet or not. That makes sense.
And cycling of temperature?
Yep.
Gonna happen in the liquid droplet?
So what's amazing is that there's a droplet generator, so it creates, I forget the numbers now, like 30,000 or 100,000 droplets, and then actually the droplets get put into a test tube, it gets put into a PCR tube, and does the normal cycle, and it gets put back into the counter itself. Yeah. So this is a really kind of emerging field. There are more companies that are getting more efficient technologies, and this is very attractive because, especially in diagnostics, because you get a real digital, real sense that comes out. Okay. Next we'll talk about DNA damage in PCR fidelity. Okay, so typically in PCR, you're right, you start with one molecule, you have a forward and reverse primer. After the first cycle, you go to 2 molecules, and then 4 molecules, 8 molecules, so it's a beautiful exponential amplification. The problem is that what happens if your starting material has a site of DNA damage? So DNA is damaged, again, your comments are what happens in the cell. DNA is damaged all the time, right? So going out into the sunlight, your DNA is damaged. Breathing oxygen, your DNA is damaged. Water can cause bases to fall off. So there's a lot of problems with DNA damage, especially if you're trying to do PCR for forensics, like a blood sample or ancient DNA. There's a lot of DNA damage. So DNA damage in the template strand, when a polymerase comes upon a site of DNA damage, it just stops, and it's inhibited by DNA damage and blocks extension here. So this is an example of a PCR reaction that this is a PCR reaction, and the DNA was exposed to UV. And so if there's no UV, you get a nice PCR product. But if it's exposed to UV light, there's damage in that DNA, and you get no PCR product. This is a problem. So in the cell, there's kind of some amazing numbers of people who study DNA damage. So there's all sorts of damage happening in the cells as we're sitting here. Apurinate sites are when the bases fall off. That happens between 1 and 50,000 times per cell per day. Deamination happens when C's get deaminated to U's. That happens between 2 and 10,000 times per cell per day. There are mismatches. Pyrimidine dimers happen in double-strand breaks, circulating chemicals, cross-links. These bulky adducts, 8-oxyG is an oxidation product that happens 10,000 times per day. Single-strand breaks. So in your cells and in your lab experiments, DNA damage is happening all the time. In our cells, there are robust, in every cell, in every organism, there are robust ways of detecting and also fixing DNA damage. So again, trying to think about enzyme discovery and being inspired by nature, trying to understand how nature deals with all these damage problems is what scientists did at Biolabs. So they looked at this DNA repair pathway called the base excision repair pathway. And this is, again, an enzymatic way of sensing and repairing DNA. So what happens is that the cytosine can lose an amino group and then get deaminated to U. Okay? So the problem with that is that if then replication occurs, this U will base pair with an A and cause a mutation. So in the cell has to sense those U's that get deaminated and fix them. So the way this works is that there are enzymes called uracil DNA glycosylases that's constantly scanning the DNA for uracils. It'll recognize that uracil. It actually flips out the base and cuts off the base. And then another enzyme will recognize this sugar without base, cut that off. And then a polymerase will incorporate a C, a correct C across from G to repair that. So if this is happening like tens of thousands of times in every cell every day, these are incredibly robust repair pathways. So the researchers at Biolabs said, hey, we understand this pathway. We can actually design a set of enzymes that will recognize and repair our DNA in our PCR or sample just using these enzymes. So they put a mix together called PCR that's based on this base excision repair pathway. They have a bunch of repair enzymes that will recognize different sites of DNA damage and a DNA polymerase and a lipase. So in the test tube itself, the combination of these enzymes will actually go into the test tube, scan the DNA for any sort of damage, and actually repair it. So what that looks like is, again, these are UV examples. So you treat the DNA with UV, no PCR product, then you repair it, you get a nice PCR product. And that happens, believe it or not, just heating up DNA causes the bases to pop off, and that's a problem as well. So again, repairing heat-treated DNA, oxidated DNA will restore the actual PCR. And why this is important we'll talk about DNA sequencing tomorrow, but as people are trying to analyze single molecules by DNA sequencing, if there's damage in that single molecule, then the sequencing will fail. So a lot of sequencing methods actually, especially for very low amounts of clinical materials, they want to repair everything beforehand so the results will be better. So that's a major use for this sort of mix of enzymes. Again, trying to understand how nature does it, what are the pathways there, what are the enzymes that are involved, and then putting them together in a test tube that will do it really efficiently. Does that make sense? Okay. So diagnostics. I think Steve talked a little bit about LAMP. This is a technology called colorimetric LAMP that was developed at Biolabs, and kind of like PCR, the specificity is from specific target-specific primer annealing. In LAMP, there's a crazy scheme of primers holding back on themselves, creating loops that get extended, that create more loops, that get extended and create more loops. So it's a crazy exponential system that has high specificity because of the design of the primers here. So there's some great kind of cartoons and movies online about how it actually works, but it's basically a way of amplifying a small amount of material to a large amount of material that you can actually detect. thing about, DNA synthesis and DNA reactions, from your high school chemistry days, is that when a polymerase incorporates a nucleotide onto DNA, it extends the DNA by one nucleotide, but it also releases a pyrophosphate from the nucleotide, as well as a hydrogen ion in the reaction itself. Okay? So, as DNA synthesis is going along, it's releasing all these hydrogen ions. So from your chemistry equations, the pH is equal to negative log of the hydrogen ion concentration. So the more hydrogen ions you have, or the more synthesis that's occurring, there's more hydrogen ions being produced, and the pH actually drops really high. So during a polymerase reaction, the pH, hydrogen ions being produced and the pH drops. Okay. So, they took advantage of this feature that some dyes change color with pH, right? So they actually showed that this dye, I forget what it's called, but it's when the pH is high, it's pink, and when it's low, it's yellow. So just by including this dye in the actual lamp synthesis reaction, you can monitor whether the target is there or not. So I have a little movie here, you can kind of see the two, it goes from pH 9 to pH 6, just because the polymerase is synthesizing and the hydrogen ions are being released. So, one more time. Starts out as red. This is sped up, of course. And then it goes over. So it's a pretty neat, and so this is a diagnostic test that you can see what's going on, right? So positive would be yellow and negative would be red.
You still need thermocycling for that to work?
So you don't need thermal cycling, so it's called isothermal, so you do it at one temperature, you do it at elevated temperature. So this is an example of like a positive, so the dye is called phenol red. Positive result here is yellow, and negative result is green. So it's pretty digital. Yep.
Is there a way to distinguish non-specific products?
That's so that has to be done by the primer design. So some primers you get a lot of non-specific, which can be a false positive, which are a big problem. Right? So that can be definitely a big problem. Yep.
So phenol red is used in like cell culture media. So is that the reason as cells age and proliferate, it's the polymerase?
I don't know. Yeah. Does it turn yellow for black? Oh, it does. Okay. The pH goes down. So
Okay. I think it turns more pink.
Okay. Okay. Yeah. I don't know.
Hmm.
So what I like about these sorts of things is you have scientists thinking about, okay, we have this lamp reaction, we have this chemistry with hydrogen ions being released, we have pH dropping.
Mm-hmm.
Okay. Is there a simple way of like figuring out what a simple readout would be?
Mm-hmm.
So connecting those dots. And the initial experiments they did was they were on pH paper. So they just dropped the PCR out of the lamp reaction on pH paper. And then they had the idea, maybe we can use these dyes, sensitive dyes to actually get the color change. So there's a lot of like, in science, there's a lot of like connecting the dots, like talking to your colleagues about what they think. So, and that was a good lesson to it. So they have these colorimetric lamps for all sorts of different applications, but during COVID, just a few weeks after the sequences were out, they collaborated with some people in the Wuhan Institute for Virology to see if they could actually develop a lamp test, a colorimetric lamp test for COVID. So that was really one of the very early COVID tests that would take advantage of this simple color change. They had another good example where there was a student who did a project and won a competition competition to send colorimetric lamp up to the International Space Station, so it can be run in space, so, and the astronauts can interpret the readout, so, kind of neat. So the advantages here is that it's about as simple as you can get, right? The print is fast, there's simple color readout. They even adapted it so you can, during COVID times, you can actually take a spit sample, put it directly in the reaction, and get a readout. There's no separate RNA purification, it's cheap, there's no instrument, it's really testing you can do anywhere with a little heater. But one of the disadvantages, it's really besides background, is that it's really a yes or no answer. So you don't get a quantitative result, you don't know how many copies of whatever you're looking at is there. It's more of a yes or no result. But this colorimetric lamp is really the basis for a lot of the COVID tests during COVID. This is a company called Lucera, and this is a really neat assay. So we broke it open and take a look at like what's under the hood here. So this is your nasal swab that you put in some buffer, and then this device here is a heater as well as a colorimetric lamp device that would give you a positive or negative. So when you break it open inside, you have this electronic piece. Can anybody guess what this electrode thing is? Yes. Yeah. Pretty neat. I didn't realize that. So on top of that heater, so a lamp happens at about 60 degrees. On top of that heater is a cassette, and you can see this cassette is kind of arrayed like a like a flower. And in each of these little lobes here, there are these little beads, and in those beads are actually dried enzymes with dried primer and dried dye. Okay? So what happens is that you put your sample in here, press it down, that liquid hydrates the little dried beads. On the heater, the lamp reaction happens. And if it's a positive, in this case it'll turn yellow, and if it's negative it'll remain purple. And so they use a slightly different dye. And then they have different LED meters here that would read out negative and positive. So it's the same principle, same chemistry. It's just miniaturized, put in dried, what I call lyophilization beads, put in a little cassette on top of the heater and LED sensors, all at the same time. So it's really pretty neat, right? So again, this is looking for a color change or positive.
And is this repeatable to make sure?
I believe there's, I'm not entirely sure how it's designed, but certainly with all these diagnostics, you have to have a positive control and a negative control. So presumably some of these chambers are controlled will probably be heated as well. Yeah. Yep.
Might be a little in the weeds, but do they actually have pumps to move the fluid around inside each of those reaction chambers, or is it just all the diffusion beds?
It's all the diffusion beds. When you push down on the tube, it clicks and it punctures something and it's all diffusion beds. This is a really cool technology, right? This is about $60, maybe more per test. And this is a one-time use device that was battery powered. So beautiful engineering, beautiful enzymatology. It really had its moment during COVID, and then the company ran out of money. And they sold the company to Pfizer, which is hoping to develop it as a flu test. But the moral of the story here is that these diagnostic tests are cheap. Right? It's $60 or $100 test during COVID, during the pandemic is one thing, but no longer in the pandemic. So it's a beautiful piece of instrumentation. There's another one called Q Diagnostic that had another similar sort of technology. They even had a Super Bowl. Now they're out of business. Anyway. So really, for any diagnostic test, there need to be rapid, specific tests for COVID-19 or any future virus. It needs to be rapid, cheap, easy to analyze, no instrumentation. So thinking about how to use enzymes for these sorts of chemistries, I think really is important, and now that you have knowledge of enzymes from this course, you guys can go out and love going to tests and make a difference in the world. But ideally, like, you test, take a picture with your iPhone, get the results, right? So people are used to doing it. After COVID, people are very much used to doing tests at home. So I think it's here to stay. Just the price point has to be right. The ease of simplicity has to be right. So there's room for growth for sure. I did want to just touch a little bit about how enzymes are absolutely critical for making the RNA vaccines, and your knowledge of enzymes now is really plugs into that. So this is kind of an older video from Moderna, but then we'll talk about how enzymes are actually used to make the RNA vaccines. Okay.
Moderna are pioneering an entirely new way of making protein therapies by using messenger RNA to trigger the body's natural processes for protein production. Known as messenger RNA therapeutics, this novel approach has profound implications for treating a wide range of diseases that cannot be addressed with existing technologies. Messenger RNA carries genetic information from DNA in the cell's nucleus to ribosomes in the cytoplasm, where it acts as a blueprint for protein production. Building on recent advances in messenger RNA biology and biochemistry, Moderna's scientists discovered a way to manufacture messenger RNA using nucleotide analogs that evade the innate immune response and effectively tap into the body's natural protein-producing machinery. Messenger RNA therapeutics can be encoded for virtually any known protein, and then specifically designed to be taken up by the cells in different tissues and organs. They can produce intracellular proteins, which remain inside the cell, as well as transmembrane proteins, which attach to the cell's surface, and secreted proteins, which are released into the bloodstream. Because existing methods for producing protein therapies are limited to transmembrane and secreted proteins, the newfound ability to produce intracellular proteins promises to greatly expand the diseases we can treat. This remarkable breakthrough also promises to dramatically speed the development of new medicines and reduce the costs of developing and manufacturing protein therapies. Moderna's messenger RNA therapeutics have the potential to usher in a new era of medicine, transforming the way drugs are developed and diseases are treated.
Okay. All right. So, the basis for RNA therapeutics is really having a template for your target and then making modified RNA with a cap. So, easier said than done. But I think knowing what we know about enzymes, you can follow the logic here. So if there's a new pandemic and a new virus that they need to make an RNA vaccine, you can see how you can do that very rapidly. So basically with gene synthesis, you can synthesize any piece of DNA and then use PCR to amplify that piece of DNA. Right? And then for RNA, you need untranslated regions on 5' to 3' end. And those are usually added by ligation with a little sequence here, and then a second set of PCR. Okay. So in this example here, we use after proofreading. Proofreading, right? Exactly. So especially if you're trying to design an RNA therapeutic and RNA medicine, you want to make sure that RNA medicine is absolutely the highest fidelity ever, right? So you don't want any mutations to be introduced there. So with the second round of PCR here, you can then clone that, sequence it, and then add a 5' cap and a 3' poly A tail just by PCR. Okay. So you can imagine that this process to generate a template for in vitro transcription, how long do you think that would take you to do? Maybe a day? You could also, because PCR is so robust and simple, you can also test 10 different templates or 100 different templates really quite easily. so once you have this, complete template, PCR product, then you can do the, use T7 RNA polymerase to do the in vitro transcription with the template that you just made, nucleotides, modified nucleotides, a 5' cap, and T7 RNA polymerase. So, you know, this is quite a simple method to make RNA, and you know, so this is essentially what they did with COVID, and of course on an absolutely massive scale. But yep.
Why is this technology in the right situation?
Well, so it's been around a long time, right? So it's been, so there wasn't an emergency need to do it. You know, at Biolabs, we were working with them for 10 years before that, and happened to build a GMP facility for these sorts of enzymes right before the pandemic. So it really needed to test. So the vaccines are one thing, but you know, with RNA therapeutics, what people really want to do is to have RNA therapeutics that will do gene replacement or you know, have chronic use or vaccines or cancer vaccines to target specific cancers. So that's really the big market. There are lots of technical problems with this. One of them is double-stranded RNA for T7 transcription. You hope to get just a single-stranded piece of RNA. Sometimes it can pull back and actually create a double-stranded piece of RNA, and that is a real signal to your immune system that something's improving. So there's lots of manufacturing steps talking about quality controls and purifications to remove the double-stranded RNA. So there still needs to be innovation around all these technologies here. But you know, doing this process here, after making RNA, treating with DNA, so this enzyme to get rid of the DNA template, phosphatase removes all the excess nucleotides. This whole process, right? This whole process is just enzymes, right? Or not just enzymes, but our awesome enzymes, right? And so I think like from what you learn in this class, what you've done with enzymes, like this should be a new grasp. Yep.
You also use something similar to make siRNA like Mylan did for the enzyme.
Those are mostly chemically synthesized, as I understand. So short RNAs, you can, like oligonucleotide synthesis, make them chemically just because they're short. Is what you say. Yeah.
But hopefully making the RNA, we need the one at all.
Yes. Yeah. So right. So making the messenger RNA is one thing, but the lipid nanoparticles put it in and then deliver it is a whole other area of science. Yeah. Super important for sure. Okay. So just to kind of end with now that you guys are experts coming towards the end of the course, you're experts on enzymes. I think it's just a reminder to be a critical reviewer of technologies for yourself and your family. This is one of my favorite examples. It's from a company called Base Pause. And at Biolabs, everybody in the company once a month would have to take technical calls from anybody who called up. And so the phone would ring, and you never knew who you're going to get on the other side of the line. So it's mostly fun and like helping people through the problem. It's great. But this company called up and had some questions. Base Pause. What the heck is that? So it's actually a consumer test, like 23andMe for your cat. And they had like some incredible like videos of how do you get a DNA sample from a cat, right? So it depends on the personality, but the cat in their videos was like the most docile thing. And they give you a piece of tape that you put on the cat's fur, and then you remove the tape and remove the fur, and then you take a saliva swab, and this cat was like, holy joke. Does anybody have any cats who would endure that? Yeah.
Yes.
So I'm much,
yeah.
how much would you pay for a genetic test for your cat?
20.
$20? Nothing. $99. Okay? So, you know, whatever, you wanna know the genetic, basis of the cat, that's totally cool. There's another one called Bino. So, this is, your DNA guide to wines you'll love. So this is another consumer genetic test that says, okay, receive your kit, share your DNA samples, and then we combine your DNA analysis with your taste preferences to reveal your Bino, your unique wine preference profile. We'll let you know when your results are ready to go. So with this one, you give your DNA sample, but then you also fill out an extensive survey about what kind of wine you like. So, I know so it's kind of crazy, right? So, yeah.
Are they still in business? Just
I
curious.
don't know. Yeah, exactly. So,
Oh
yeah.
my gosh.
So many cool technologies out there. There's so much creativity in terms of molecular biology, discovery, and using enzymes in really creative ways. But just, keep an eye out. And of course, the big one here is Theranos. If you haven't read the book or seen the documentary, it's really fascinating. And I think wishful thinking is never a strategy, right? And I think just being critical reviewers of all these technologies. And this was really ahead of, the concept was ahead of the time where you can take a droplet of blood and have all, results from all sorts of chemistry. Still not there yet, and I think that is the goal, to really understand our human health and disease from small samples, and that really is the trend. But yeah, so that's about it. Tomorrow we'll go over DNA sequencing, and again, the whole basis of all these new DNA sequencing technologies is enzymes, and there are constantly new sequencing technologies that come out